Hi, everyone. Yes, I am Brook Santangelo, and I'm in the Computational Bioscience PhD program at CU Anschutz. And I'm going to talk about, again, the influence of the microbiome in disease today. So microbes account for a huge amount of diversity in our environment, and that genetic variation enables them to adapt to a range of environments. And so I'm specifically interested in the human microbiome. And this human microbiome has many different environments that allow that are different niches that microbes can adapt to. For example, in the skin, you have a majority of facultatively anaerobic bugs versus in the gut, most are strictly anaerobic, and they are able to thrive in that environment. They play a critical role in human health. And microbes are able to, for example, metabolize what humans otherwise could not. For example, break down fibers into short-chain fatty acids. That helps modulate our immune system. And they also maintain a balanced environment, such as in pH in specific places. So we're interested in what makes this microbiome become dysbiotic, and why does that happen, and how. So to understand the microbiome, we go about studying this through a systems-level approach. We have to understand many different data types: metabolomics, transcriptomics, gene expression, in addition to the microbiome data, in order to fully understand their influence. Microbes are able to be modulated in many different ways. So we understand, for example, in the environment, such as stress or changes in sleep habits can greatly alter your gut microbiome in negative ways. Of course, drug treatments, such as antibiotics, that can wipe out the positive commensal bugs and result in, for example, Clostridium difficile infection. And there's also this feedback loop between host physiology, diseases, and the gut microbiome. And to really start to understand how the microbiome can be modulated in order to effectively and safely treat these diseases, we have to understand and approach this from a systems level. So this leaves us with a gap in how we study the microbiome currently. We will first, in order to understand the influence of a particular microbial environment in a disease, we'll first perform sampling, and picking the right community or population of disease or healthy controls is difficult to alleviate confounding factors or other factors that may influence that choice. We then sequence that microbial environment, and we're now with sequencing able to do this using 16S or metagenomic sequencing, and then we can then classify from a relative and understand a relative abundance of which microbes thrive in the diseased environment versus the healthy environment. And this leaves us with largely associative results. So we can understand that a given bug or a given environment of bugs is significantly over or under abundant in a disease population versus a control. We're starting to get at the functional annotation question, as we talked about in the last talk, that allows us to start to tease away at these molecular mechanisms, but to really understand how this community of microbes is influencing disease. These are the questions that we're trying to answer now. And so in order to do that, there's a lot of knowledge out there that exists in papers and free text, in microbial experiments or gene expression experiments of the host, or organized databases. And it's critical to integrate these and harmonize this data so that you can start to query it in a way that we can perform mechanistic inference and get at specific molecular explanations for exactly how a microbe or a community of microbes may influence a disease. And so we've started to do this by looking at the information that is out there that is computationally readable. And we broke this down into 6 different categories of knowledge. Of course, we've all talked about ontologies or taxonomies, which are systems that are used as a semantic standard and describe the hierarchical relationships between a given field or a group of fields. And then we also can incorporate annotated databases, for example, which store experimentally derived content. And it's important to then be able to incorporate these correlative curated databases. And these are those that provide assertions of associations, for example, between microbes and disease. What's really important for us to be able to get at the mechanistic inference question is these semantically consistent knowledge bases, as we've named them, or knowledge graphs as we all know them. And these are able to start to integrate in a semantically consistent way these different classes of knowledge. For example, microbes pathways, reactions, metabolites, and diseases. And we need more of these in order to perform this question of mechanistic inference and understand mechanisms that explain the associations that we know. And so we introduced this modular knowledge graph construction framework that is relevant in this microbiome context. This integrates functional annotations, genomic traits, and overtop these open biomedical ontologies using the KG Hub framework. And it uses the BioLink model, which is a standardized way of representing biomedical entities such as genes, diseases, chemicals, phenotypes, etc. And this allows us to then also align with this larger KG ecosystem, for example, with the Monarch Initiative or NCAT's Translator Project, so that we can further ingest other resources given different use cases that we may have in the future. And for the purposes of this talk, I'm going to talk about 3 of these resources, starting with KG Microbase, where everything is built off of, and this new knowledge graph, KG Microbiomedical or KG Microbiomedical Function. And each of these have different sizes and increasingly ingest more information as you go down the line. And so we have these kind of catered to different use cases, such as if your question is more around environmental applications, or we incorporate new health and disease-focused resources for KG Microbiomedical to start to answer more of the mechanistic inference questions about the microbiome in disease. And so I'm going to discuss a couple of the modeling choices that we made along the way to integrate these resources. And first, in order to model microbial traits, we utilize the bacterial metadatabase BacDive. And BacDive provides this standardized prokaryotic data at the strain level based on experimental data and observations. And it provides this in many different classes. So you can get morphological information as well as metabolite utilization, etc. We also integrated microbial trait data from this Madden et al. Paper, which provides a machine-readable trait database that covers 14 different phenotypic traits and other quantitative genomic traits, environmental characteristics, etc. And we were able to model that and map that to many of the ontologies that we can then provide more of that hierarchical structure and semantic structure. And so this is an example of how we were able to then represent the relationship between organisms and the chemicals that they produce or consume, the actions that they're capable of that we aligned to enzyme commission numbers or Go molecular functions, and then a couple other new entity types that we introduced, such as isolation source or media or cell shape or other cell characteristics. And then one other example of this is how we modeled chemical reactions in participants. And we did this using the RIA database, which is an expert curated database of biochemical reactions. And we were able to base this on the Kebi ontology. And we were then using this sort of subclass of schema, we're then able to represent both the bidirectional reaction that is connected to many other databases, such as the UniProtein database. And then we can also connect those chemicals in ways that describes that relationship. So whether they are input or output of which direction of the reaction. So knowing this, how can we utilize this resource now to then understand why an association may exist between the microbiome and disease? And so we chose to do this over inflammatory bowel disease. And inflammatory bowel disease encompasses 2 different conditions, ulcerative colitis and Crohn's disease, and it generally is implicated by chronic inflammation along the small and large intestine. And it's generally known that that the gut microbiome has a large influence on this, though the specific mechanisms at play are largely unknown. And so we know that the microbiome is dysbiotic, that the balance between commensal microbes and pathogenic microbes is in flux, but the exact mechanism, again, is unclear. As you can see in this bottom panel, it's generally characterized by increased intestinal permeability that allows for more inflammation in the intestine from pro-inflammatory cytokines, etc. And so in order to query this knowledge graph to see how we could start to get at these mechanistic explanations, we utilized the KG Microbiomedical graph. And we use the organismal traits that I described earlier from the BacDive database in the Madden et al. Paper and represented them as this binary feature table and used training on an external data set that describes these microbe disease associations. The DISC-Biome database is one of those that describes these interactions, where they gathered many different PubMed publications that found some correlation between a specific microbe and a disease. And so we were able to find 109 associations between organisms and IBD, ulcerative colitis, and Crohn's disease. And we looked at this using gradient-boosted decision trees on how well this model was able to find microbial traits and features that may influence the outcome, that is likelihood of inflammatory bowel disease. And so what we found was some really interesting biological results. I highlighted 4 key metabolites that came up as the top features in this model in its influence of predicting this outcome of increased or decreased IBD. These 4 metabolites, D-mannitol, maltose, raffinose, and xylose, are small fermentable metabolites, or FODMAPs, that have been shown to exacerbate IBD symptoms of bloating and abdominal pain because they increase gas production in the gut and they're poorly absorbed. And so this suggests that the microbes that are able to consume or metabolize these small fermentable metabolites are better adapted to thrive in this gaseous environment that IBD may create. And while it doesn't yet get a causal direction of this interaction, it starts to allow us to understand which organismal traits are important in this context. We also see several examples of environment pointing in different ways on the influence on IBD, such as mouth, skin, and vagina environments that contain bugs less adapted to the intestine versus that suggests that the pathogenic bugs that we mentioned earlier are better adapted to thrive in this environment. And while we don't see great performance in this model, the question really is about what important biological features may be interesting, and we're starting to get at these mechanistic questions. So next, we started to ask whether we could actually use the ingests that we had represented semantically in the graph to help us with this performance as well as with the features that we ended up choosing. So we also incorporated in the feature table strains that were subclass of species that may be in that DISC-Biome set, but not annotated to the trait, even although their strain may be annotated to the trait. So this increases the training data that we could have in this model. And what we found, what we found was that we got a couple of those new features that we incorporated into the model as some of the top features in predictive, in how well we could predict likelihood of IBD in which direction, and that they are relevant to the disease. So we have capable of nitrate reduction to ammonia, being a human pathogen, as we talked about earlier, which is relevant in the IBD context, and having the phenotype of being microaerophiles, which we see 2 genera, Helicobacter and Campylobacter, which have been implicated in IBD and are microaerophiles. And although we're not only looking at the performance of the model, we do see an increase in that by incorporating the semantic structures that we've now been able to introduce into this knowledge graph. So this leaves us with the question of how can we then also incorporate the genomic annotations that we've introduced into this same question of understanding the associations in the IBD context. And so I'm not introducing those results in this talk. I did want to talk a little bit about how we modeled the genome annotations in the knowledge graph. We did this using the UniProt database, and we focused on the SwissProt or reviewed proteins. And we took only microbes that have a reference proteome that exists in the database. So those that have, of course, been more studied, but have more of this information in the database. And we were able to grab a large range of different classes of categories of knowledge. And so we are able then to describe how an organism expresses different genes, the cellular components and molecular functions that they're involved in, and then the reactions and enzyme commission number in EC numbers that those are associated with, as well as the chemicals aligned to KEBI that they bind to. And so this can really help us in the future with this model to understand functionally how those features may be influencing the associations with the disease. So in conclusion, we developed a framework to build these customized microbiome relevant KGs, and I just presented a few examples of these graphs that can be used for this purpose, where we're able to integrate functional annotations, organismal and genomic traits And also we provide a detailed modeling structure for modeling pathways and reactions. We can also, in this framework, select a subset of taxa given for a specific use case so that the graph isn't getting too big and hard to manage. We also showed that this harmonization of these different data sources enables us to start to get at this mechanistic inference question in this IBD use case. So in the future, we'll apply these functional annotations that I showed that we've done from UniProt and the graph in the disease context. And then we'll be able to expand this mechanistic inference to also include template-based path searches so that you can really get the molecular explanations for specific bug to disease associations. So with that, I just want to thank my collaborators at Lawrence Berkeley National Lab, Martine and Harshad, and then my advisors, Dr. Larry Hunter and Dr. Kathy Lozapone, and of course the organizers for inviting me here, and I'll take any questions. And if you're interested in looking at the past publication of KG Microbe, you can look at this one. This work is in the works for a future publication. And you can also take a look at the GitHub for this framework.
Thank you. I enjoyed that. You're trying to bring in a lot of information together, and I'm glad you've got this case study disease to try and explain. I was trying to see where time came into this, because you're talking about mechanistic approaches, and you do talk about reactions and pathways. How else do you capture time? How else do you capture the changes that happen, like even the predator-prey model within the microbiome, as to who dominates? And then how do these attack the host? How do they get through the cells into the gut and have their effect? So are you planning to do anything with time? Are you planning to do anything with dynamics? Because ontologies are still pretty much static.
Mm-hmm. Yeah, thank you for the question. I think it's a really good point, and that I haven't incorporated, of course, into this resource, and I think there's a lot of potential not incorporating that yet. But I will say one thing that this allows you to do is you could sort of build this subgraph that is focused in on a particular cell type, or a particular, or even a particular time-based data set that you could base it off of. And the model of how we built this resource allows us to then start to ingest that and focus in on that further. And so in this context, I've really focused on a use case that is kind of this broader, higher level features that influence a disease. But I think that you have an excellent future direction for how to kind of start to use this framework for those kinds of questions. So
thank you very much. Very, very interesting talk. So I have one question, because a lot of your analysis relies on prior knowledge about functions in these microbes. But I see two problems with this, because we know about 60% of the proteins don't have any functions in these microbes. And as we just heard in the previous talk. And then the second is, I don't see where you use genomic data or metagenomic data, because you said you get 16S data, but from 16S data, you really just call maybe a species, and then you use some public genome that has been seen. But these microbes, they have plasmids, there's horizontal gene transfer. So there's a lot happening that you do not actually see if you just rely on prior knowledge and existing functional annotations. Do you have any idea how you can incorporate metagenomics data so that you can see what is actually present, at least in what is the metaproteome that is present there, that could exhibit any function?
Yeah, so a future way of representing this graph is by incorporating, so a lot of this is based off, we said for now, the UniProt reference proteome, the Swiss Prot database. And so we're largely basing everything off of what we know in that sense. We aren't using the 16S data in the graph. We were using the DisBiome set for this as this test data set to try to understand those associations. But those edges are quite different than those that are the functional edges describing this protein does this thing and is involved in this reaction. But in terms of incorporating metagenomic data, one thing that you could think of doing is, rather than characterizing by NCBI taxonomy or like naming them by their name, you can start to incorporate their full genome, or take from these databases like RefSeq or GTDB, and you could represent them as that sequence as opposed to the name of what we've classified them as currently, their current phylogenetic classification. And then all the features of that genome then become what describes different reactions, go processes, et cetera. So again, I think it's sort of where we're at with this analysis, is taking what we know maybe obviously and then moving towards incorporating more, drilling down into the different genome characteristics that we may want to model in the future. Yeah.
You mentioned that you created this modular knowledge graph building platform or framework. I'm wondering how it compares to the existing modular knowledge graph building models, such as MOMO and NARM.
What was the one you said?
MOMO and NARM.
Okay, I don't know that I know that one. So we are kind of in this phase of trying to think about how to, this is really built off of the KG Hub framework within the Monarch Initiative. And so it's kind of figuring out how to utilize all of these tools within KG Hub to perform these ingests. And then what kind of unit of knowledge that we've ingest to make available, so that it's sort of, if you think about it, you're pulling different pieces of, like, we have the UniPro piece and the RIA piece, et cetera. And so it's largely, I think that there's a lot of room to, for example, the one that you said, to start to try and like talk more about how these knowledge graphs are being created, so that they're not all in a silo and we're not all ingesting the same information in different ways. I don't know if I answered your question fully.
Okay, any other questions?
Nobody else does. I can speak a little bit to your question. So I think Brooke's claims are really largely about knowledge modeling. And she's using existing systems for building the graph out of that modeling. But it's really semantic choices about what gets modeled, how it gets mapped onto BioLink and things like that. The tools that she used to actually build the knowledge graphs are mostly not hers. And so you could imagine using the same modeling decisions, the same basic ideas for sources, and with different underlying frameworks if you have a reason to do that.
Right, yeah, no, it's just, these are different approaches to building modular ontologies, larger discussion perhaps, but I just wanted to see if you kind of knew about it and how that